is Biomed Cafe menu. Bacterial Pili will be presented today. We will talk about Pili discovery, Pili structure, Pili function, and quiz game and puzzle. I'm Dr. Biomed, cellular and molecular biologist. Hi everybody, welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified, and summarized form. Pili discovery. They saw something others didn't see. Charles C. Brinton in 1959 discovered a feature of bacterial cells that he called pili, describing them as non-flagellar appendages. Brinton in 1974 proposed that injecting harmless pili into the body could elicit peeling-specific antibodies, thereby preventing many diseases of bacterial origins. Pili characteristics. They are a hair-like appendage. They are found on the surface of many bacteria and archaea. They can be considered as an antigenic structure. They are fragile. They are composed using oligomeric peeling proteins. Some of them are not membrane-bound organelles. Some of them are not motile appendages. Some of them are dynamic structures capable of extension and retraction. They are about 6 to 7 nanometers in diameter, and they are about 0.3 to 3 micrometers in length. Pili types. Pili can be classified by two options: structure and function. In classification by structure, pili can be divided into two groups: gram-negative bacterial pili and gram-positive bacterial pili. Gram-negative bacterial pili can be divided into 5 subgroups: Chaperon-usher pili, type 4 pili, type 4 conjugative pili, type 5 pili, and Curli fibers. Gram-positive bacterial pili can be divided into 2 subgroups: fibrils and pili. In classification by function, pili can be divided into 2 groups: conjugative pili and Fimbriae, Chaperon-usher pili. They are widely expressed among type of gram-negative bacteria. They have 2 parts in their structure: tip fibrillum and rod. Usher is an assembly platform. Is involved in pilus biogenesis. P-type and type 1 pili can be distinguished by their usher. They play a role in host attachment and biofilm formation. They can be divided into 2 subgroups: type 1 pili and P-type pili. Type 1 pili. They have 2 parts in their structure: pilus rod and tip fibrillum. Pilus rod. Its thick and helical cylinder. Its components are Fimbriae, about 1000 copies. Tip fibrillum. It's thin and short. Its extent from rod. Its components are major and minor. Major components are Fim F and Fim G. minor components are adhesive proteins or adhesins or Fimh. They are located in the outer membrane. Example of a species is E. coli. Type 1 pilus biogenesis is by Chaperon-usher system. They play a role in adhesion, aggregation, immunomodulation as biosensor and biofilm formation. P-type pili. They are called P. pili or PAP2. They have two parts in their structure, pilus rod and tip fibrillum. Pilus rod is thick and helical cylinder. Its components are PAP-A, more than 1,000 copies. Tip fibrillum is thin and short. It extends from rod. Its components are major and minor. Major components are PAP-K, PAP-E, and PAP-F. Minor components are adhesive proteins or adhesins or PAP-G. They are located in the outer membrane. Example of a species is E. coli. P-type pilus biogenesis is by Chaperon-usher system. They play a role in mediating adherence to host cells and pathogenesis of urinary tract infections. Type 4 pili. Characteristics. They are dynamic structures. They involve in bacterial motility. They can rapidly elongate and retract to create the mechanical forces that allow bacteria to move. They are tube-like structures. They are strong structures and also flexible. Example of a species in some gram-positive bacteria is Clostridia, and in the majority of gram-negative bacteria are E. coli, Neisseria, and Pseudomonas. Type 4 pili have two parts in their structure, filament and subunits in the membrane. Filament has 1,000 copies of pilin, as tip pilin and major pilin. Subunits in the membrane are secretins and core proteins. Secretins form oligomeric gated channel for secretion of pilus subunits. They are located in outer membrane. Core proteins are located in inner membrane. Rapid extension and rapid retraction generate enough mechanical force. Then it pulls bacteria together. Finally, sharing of genetic material in a process driven by ATP hydrolysis occurs. Type 4 pili biogenesis is by type 2 secretion system. They are located in both in the inner membrane and outer membrane. They play a role in adhesion to surface and cells, aggregation, immunomodulation, motility, DNA uptake, and biofilm formation. Conjugative type 4 or sex pili or F pili. Characteristics. They are dynamic structure. They are tube-like structures. Their diameter is 7 nanometers. Their length is 20 micrometers. They are strong structures and also flexible. They are called sex pili because they allow for the exchange of genes via the formation of mating pairs. They are encoded by the F plasmid. They have two parts in their structure, filament and subunit in the membrane. Filament has 1,000 copies of pilin, as tip pilin and major pilin. Subunits in the membrane are secretin and core proteins. Secretins form oligomeric gated channel for secretion of pilus subunits. They are located in outer membrane. Core proteins are located in inner membrane. Rapid extension and rapid retraction generate enough mechanical force. Then it pulls bacteria together. Finally, sharing of genetic material in a process driven by ATP hydrolysis occurs. Example of a species is male gram-negative bacteria. They are located both in the inner membrane and outer membrane. They allow the transfer of DNA between bacteria through the creation of mating pairs. Type 5. They are unique to gram-negative bacteria. They have three parts in their structure: tip, stalk, and anchor. They consist of pilin proteins. They are located in the outer membrane. Example of a species, Bacteroidea. Type 5 pilus biogenesis is by protease-mediated polymerization. They can be divided into two subgroups, major or long pili and minor or short pili. Major or long pili is between 0.3 to 1.6 micrometers in length, but minor or short pili is between 18 to 120 nanometers in length. They play a role in adhesion, aggregation, and biofilm formation. Curli. They have three parts in their structure: filament. Filament has a lot of CSGA. They aggregate and form amyloid fibrils. Nucleator. Nucleator or CSGB is required for the nucleation and organization of CSGA and other subunits. They are located in the outer membrane. Example of a species, gram-negative bacteria like E. coli. Curli pilus biogenesis is by nucleation precipitation pathway. They play a role in adhesion, aggregation, immunomodulation, and biofilm formation. As I told you a few minutes ago, gram-positive bacteria pili can be divided into two subgroups, fibrils and pili. Gram-positive bacteria pili. They have three parts in their structure: pilus tip, pilus shaft or rod, it's made of repeating proteins or pilin, and pilus base. They are located in the outer membrane. Gram-positive bacterial pilus biogenesis is by sortase-dependent mechanism. They can be divided into two subgroups, fibrils and pili. Fibrils have short, thin rod. Example of a species, Streptococcus Gordoni, and Streptococcus oralis. Pili. Pili have longer and flexible rods. They comprise 3 covalently linked protein subunits. Example of a species: Streptococcus and Corynebacterium. They play a role in adhesion and biofilm formation. I explained about conjugative pili some minutes ago. Now I want to explain about Fimbriae. Fimbriae have some characteristics. They are also known as attachment pili. They are shorter and thinner version of pili. They are a stiffer version of pili. They are numerous in number, ranging from 100 to 600 filaments per cell. Example of a species: Fimbriae are found in gram-negative and gram-positive bacteria. They are located in the outer membrane, in the poles of the cell, or over the entire surface of the cell. They play a role in adhesion and biofilm formation. Now I want to compare pili, flagella, and cilia. Location: pili are found in prokaryotes and archaea. Flagella are found in eukaryotes, prokaryotes, and archaea. And cilia are found in eukaryotes. Number: pili are many, flagella are usually single, and cilia are one or many. Source of energy of pili is ATP hydrolysis. Energy of flagella is ATP hydrolysis or proton motive forces. And source of energy of cilia is ATP hydrolysis. Motility: pili are non-motile and dynamic. Flagella are motile, and cilia are motile or non-motile. And length: pili are short, flagella are long, and cilia are short. Key points of pili. Pili are components of the bacterial cell. They help bacteria to attach to the host organism. They facilitate the process of mating in order to share the DNA particles between the cells. And they have an important role to play in the causing of infections on hosts. Thank you for watching. Please subscribe to this channel, and if you like the contents of this video, please like it. Now it's time for the quiz game and puzzle. I have put the link to the quiz game and puzzle in the description of this video. Download it for free and enjoy it. Until the next video, bye!